Hello. Just coming over here with this bottle. You are just in time. We're about to review what we left off last time. Oh, if anybody wants to hold of this, this is just a lab list of stuff we're going to talk about. Does anybody want one of these ahead of time? Nope. You just got one of those? Sorry. All right.
Thank you.
Got one? Mm-hmm. All right. Everybody good?
Thank you.
We'll just get that out of the way for now. I wanted to start over here by doing our sketch of an EKG. I'll do this in a couple of different colors.
So
maybe.
EKG, sorry.
Yeah, what's up?
An EKG could also be called an ECG?
Yeah.
Okay.
Electrocardiogram. Yeah, they're either one. Old terms and new terms, so you'll see either one. ECG, I'll put EKG. So it graphs that electrical activity of the heart in terms of waves. So we're gonna do our electrical and mechanical events. I think I'll just put any of the electrical events in green. And remember, the electrical events, we use terms like depolarization and repolarization. And then I'll try and put everything, or all the mechanical events in red. And we use the terms systole and diastole to describe contraction or relaxation, these mechanical events. Okay, let's just draw the series of waves that we saw last time. I'll just start with the flat line, and then we'll have our first wave. In fact, I want to draw a couple of them here, just so I have some room to label it. So we'll have another identical first wave, a series of second waves, then a third wave before more flatness. The first wave is an electrical event. So let me get the right color before I do that. We have electrical events here. This whole thing is an electrical event. I'll leave the other one over there unlabeled. And any of these flat segments are going to be mechanical events. We need to know what mechanically is occurring in those areas. We'll just start here as well. First wave was our P wave. The next series, this is our Q, our R, and S wave. We'll just refer to this as our QRS complex. There's a couple of things going on there that will be noteworthy. And then the last electrical event is our T wave. I'll grab a different color here to represent our mechanical events. The first mechanical event, I'll try and bring this thing all the way over here. We said this is our PR segment. The other one between the S and the T wave, this little bit is our ST segment. And then between the T wave and the very next P wave is our TP interval. Doing good so far? Okay, so now for some questions. What did we say was occurring during the P wave? Obviously, we've got it marked as an electrical event. Without the slides down, though, what's going on there? We know the terms that we have to use, either depolarization or repolarization. Yep. Of atria, that's right. And it's both atria that depolarize at the same time. And I'll pull that little video clip down that was, kind of a basic video clip, but it was showing both atria contract at the same time and push the final 30% of blood into the ventricles before those ventricles contract. And we kept saying last time these electrical events always precede mechanical events. So that's right, the P wave is when the atria depolarize, and immediately following depolarization we see that mechanical event, which is the ST segment. What happens in that ST segment? Did somebody say, what was it?
The ventricles are contracting.
Almost. The ventricles haven't yet received their electrical signal. But what has received an electrical signal are the atria. Remember, if I stick a paper clip into this, my muscles are not contracting, but if they get hit with some type of electrical current, then they are going to contract. And right now we see the atria have been hit with an electrical current. So immediately following that, we get our mechanical event of the atria contracting. Yep, atria contracting. And that's what I was talking about. When those atria contract, that final 30% of the volume of blood is going to enter the ventricles. Okay, so this atria contract. The final 30% of that ventricular blood is going to be forced into the ventricles. And now those ventricles are completely full, and we run into this QRS complex there. Obviously, it's a big electrical event, so there are a couple of things that we need to mention in terms of electrical events. I can list them as just a 1 and a 2, maybe. That might be best. First electrical event of the QRS complex. Does anybody remember that one? I guess you can list them in any order.
Depolarization of ventricles?
Well, it is a depolarization, but we're still in the atria. And when we were graphing those electrical events last time, always following a depolarization was a repolarization. So the atria depolarize in the P wave, and one of the first things that happens in the QRS complex is those atria get the repolarization, which is going to cause them to relax. So atria repolarize. And at the same time that those atria repolarize, the ventricles are going to depolarize. So two things there. Hopefully that's clear with that QRS complex. So there's our second series of electrical events, and following those electrical events, we have our next mechanical events, which happen in that ST segment. Two things are going on here. The atria have just been hit with that repolarization. So one thing that happens is the atria relax. I could have said atrial diastole. Use those terms. And the second thing that happens is the ventricles have just received their depolarization signal. So now in this ST segment, they contract. And in the graph, I'm just remembering the slide we saw last time, you likely will see some questions either in the quiz or on the exam about the position of the valves, whether they are opened or whether they are closed, in terms of like whether the atrioventricular valves are opened or closed during ventricular systole, or whether those semilunar valves are opened or closed during ventricular systole or ventricular diastole. Again, that little video clip that we saw was showing that. Okay, almost to the end. T wave is our last electrical event, and what is happening there in the T wave, the ventricles have just been told to depolarize. They contract, and following that contraction, they're now going to repolarize. So T wave is our ventricular repolarization. Following that ventricular repolarization, the ventricles now relax. And remember there in the ST segment is when the atria relaxed. The atria have not received another electrical signal. Muscles are not going to contract unless they receive an electrical signal. So now in this TP interval, we have for a second here, both atria and ventricles are relaxed. Hopefully that's not too crowded in there. But that TP interval, what's interesting is both atria and ventricles are relaxed. And it's really during that time, this TP interval, that you have most of the blood that enters the ventricles. We said passive filling of the ventricles. And that's going to be about 70% of the volume of blood that enters the ventricles is going to do so here during this TP interval. And we can see following that TP interval, there's that next electrical signal that's going to tell those atria to contract. It's not labeled here, but we can see our PR segment that's going to force that remaining 30% of blood into the ventricles. Then we have our QRS segment where the ventricles are told to contract. They finally contract during the ST segment. Hopefully that is readable. Here we have our EKG, and it's just like the one that we drew on the board, except maybe a little more colorful. The thing I like about this one is that it shows, in addition to just words on the board that we were labeling, it kind of shows what's going on with each of those segments. So like we just wrote there, the P wave is when the atria are depolarizing. And you can see that wave of electrical current spread through both atria at the same time. And you see simultaneously both left and right atria are going to contract in that PR segment. So following that electrical event, we have our first mechanical event. Then the next event that's labeled as the Qrs Complex, we put ventricular depolarization as well as atrial repolarization. So two things are occurring right there in the Qrs Complex. Two of those electrical events. Meaning two of these mechanical events are going to occur in the ST segment. They've got the ventricles contract. But we also listed that in addition to the ventricles contracting, we've got the atria that are now relaxed. And they remain relaxed until that next P wave. So anyway, atria relaxed, ventricles are contracting, so we're in ventricular systole. We're at that ST segment, and then the T wave comes and tells those ventricles to repolarize. So we have our next electrical event. Following that ventricular repolarization, we've got both, for a short time, the atria and ventricles that are relaxed. Passive filling of those ventricles. I think this, oh, it's not plugged in. Somewhere near here is the video that I kept showing where you can see both atria contract at the same time. You can see for a fraction of a second when both atria and ventricles are relaxed, that's when most of the blood is filling into the ventricles, so that passive filling. And then right before the ventricles get all the way full, the last 30% of blood or so are forced into the ventricles by those atria contracting. So if we were in atrial systole, the atria are contracting, would the AV valves be opened or closed? Atria are contracting, they're pushing on those trap doors to open. And they would want to open into the ventricle. So those AV valves would be open if the atria are trying to contract. So during atria contraction, atrial systole, AV valves are open. We see it right there. Now in ventricular systole, when those ventricles start to contract, remember the first thing that happens is those AV valves are slammed shut. That's the first sound of the heart. So if you see some question about during ventricular systole, would the AV valves be opened or closed, then they would be closed. The valves that are open during ventricular contraction, we can see are those semilunar valves. They open easily in that particular direction, while the atrioventricular valves are closed. Then the thing that slams shut those semilunar valves is the relaxation of the ventricles. When the ventricles relax, those AV valves are opened, but the semilunar valves are slammed shut, the second sound of the heart. So you can maybe bet on some questions about in ventricular systole or in ventricular diastole, would the AV valves be opened or closed, or would the semilunar valves be opened or closed?
And ventricular systole, the AV are closed, right?
So here we go, ventricular systole, those ventricles contract. And yes, that's right, those AV valves are the first ones to slam shut. And as those ventricles continue to contract, we can see those semilunar valves open. So AV valves would be closed, semilunar valves would be open during ventricular systole. And it's the opposite in ventricular diastole. We can see when those ventricles relax, those semilunar valves are slammed shut. And then those AV valves are pulled open. That's when we have that passive feeling of things moving into the ventricles. So do I got permission to jump into blood vessels for a little bit? Okay, I'll leave that up and I'll come over here to the other side. So here's something that I'll put up. We've already talked about the heart, so I'm going to draw that small and get that out of the way. The thing that we're going to see with arteries is that arteries are always going to be away from the heart. So our major arteries, these are going to lead into, when we find these things in lab, smaller arteries called arterioles. Erase that real quick. Let's give these their real names. The ones right next to the heart, we're going to call elastic arteries. And when we look at these elastic arteries like the aorta and some of the larger ones, they have thick layers of elastic tissue so that when the heart actually contracts, they expand. And then that elastic tissue recoils and it kind of helps force blood through the blood vessels. It's kind of like an additional pump. And I'm drawing arteries in blue here on purpose because I don't want you to think arteries are always red or veins are always blue. The color just refers to oxygen rich or oxygen poor. And the name artery or vein has to do with whether it's going away from the heart or towards the heart. So veins are always towards the heart, and here our arteries are always away. The major arteries right next to the heart are the elastic arteries. They're going to end up getting closer to organs and become what we call muscular arteries. And it's these muscular arteries. Their name is there because there's a lot of smooth muscle. And it's that smooth muscle that involuntarily will constrict or dilate and determine whether blood is flowing into a particular capillary or bed or not. These muscular arteries are going to lead into the smallest arteries that we will see. We'll call these arterioles. Arterioles also are layered in smooth muscle. And these arterioles are going to lead directly into capillaries. Let me get a good capillary color. There's always exchange that happens during the capillaries. Nope. So we have our capillaries. that are going to lead into the smallest veins that we're going to call venules. Just get our road map here. These venules are kind of like tiny little creeks that lead into larger rivers. And in this case, could have given it a little more room there. These venules are just going to lead into veins. And the major veins that are going to take blood back to the heart, we said were the superior vena cava and inferior vena cava. I'll just draw these going directly into the heart. Or coronary blood vessels are going to be taking that blood directly into that right atrium from the walls of the heart themselves. So we kind of have a road map. Let's see what we can add to that. Obviously, when we're going away from the heart, we're going to be using terms like fork and diverge or branching. And when we're talking about veins, these things are going to join or converge or merge. We'll say towards. So veins always towards the heart, arteries always away. If we think about the hollow space, I'll just jump to this picture right here. You can see this hollow space inside the vessel. That hollow space we're going to call the lumen. You can see a much larger lumen on the veins than you see on the arteries. You can also see a couple of other differences here. There is much higher pressure in the arteries because they're closer to this pump. So they have a much more rounded appearance, much more pressure against their walls than the veins do. The veins are going to have a much larger lumen. There's lower pressure in the veins. You can also see these layers in the walls of the arteries or the veins. I'm going to talk about those layers of the walls in the arteries or the veins. I just want to go back to the slide I skipped over. This one, the outermost layer is going to be what we call the tunica externa. Some textbooks will use the name adventitia. This just refers to the tunica externa. It's an older name. The middle layer is the tunica media. And the innermost layer is what we call the tunica interna or tunica intima. Either one of those works. This is made out of epithelial tissue, simple squamous. It's like a slip and slide. And then we mentioned the hollow space in the middle is the lumen. The difference between these three, the tunica externa, the tunica media, and the tunica interna, changes as we go from those major arteries all the way to the veins. So I like this picture because it tries to show us something. One of the things we need to remember about the artery is that in arteries, the thickest of these three layers is the tunica media. Maybe we can list that. So the tunica media is the thickest layer in the arteries, and for the veins, it's actually going to be the outermost layer, that tunica externa, that's the thickest. You can see that somewhat if you look on the side of the slide. That vein has a much thicker tunica externa. That's one of the differences between the arteries and the veins. The other two we had already noticed that the lumen was much smaller in arteries than it is in veins. You can see the irregular shape of that lumen in the veins. We can also see a third difference here between arteries and veins, and that is that these veins have little flaps of the internal layer of cells that form these valves. So the tunica intima or tunica interna has these valves. They're more common in arms and legs. They just prevent backflow of blood through the veins. The veins don't have a pump that like the artery does. So all the pressure is on this arterial side, and then we see that we kind of lose that pushing pressure, the hydrostatic pressure, and the rest of it is, we'll see at least four things that are going to help blood move through the veins back to the heart. Keep in mind there's no pump on this side, so things like the valves and the low pressure and that lumen are going to be things that help blood move through the veins. Down here in the capillaries, there's just a single layer. The only layer down here in the capillaries is that tunica interna. So being just a single layer thick down here in the capillaries, it's going to make it easy for exchange to take place. You can see that single layer of squamous-shaped cells. There's a thin layer of basement membrane. But things that are small enough that can just squeeze in between those cells are going to leak out of these capillaries. In fact, we're going to see at least 3 different types of capillaries in our introduction today. But back to this slide, you can now see at least a couple of differences between arteries and veins. That large lumen in veins, small lumen in arteries, and that thicker tunica externa in veins, where in arteries it's the tunica media that's the thickest layer. Okay. This also just shows that as inside the left ventricle, you can see the pressure fluctuates greatly depending on whether or not that ventricle is relaxing or whether it's contracting. And these large arteries, the ones that are right next to the heart, these are these elastic arteries that are capable of stretching and rebounding. So the pressure in those arteries fluctuates as it's next to the ventricles that are pumping. But as we get further out into the arterioles, you can see the blood pressure starts to level out. In fact, in the physiology part of this class, if it was a separate physiology class, we would likely have to calculate this mean arterial pressure. Anyway, we don't have to do that. But what this is showing us is that as we go from the arterial side all the way to the venule side, there's a constant decrease in pressure because you're getting further and further away from that pump. Next to the heart, I said those arteries are called elastic arteries because they have many layers of this elastic tissue. That's helpful because it allows those things to stretch out and expand to accommodate that extra blood. And then as they recoil, that helps force that blood further down the vessels. So it kind of helps the heart out. Here are those mid-size arteries, the ones that we called muscular arteries, get that name because of their thick tunica media. And it's really these muscular arteries that determine, whether blood is shunted in a particular direction or not. If you're in fight or flight, then it's going to shunt that blood away from places like the digestive system and towards places like the blood vessels. Let me try that again. Away from the digestive system, towards the skeletal muscles is what I'm trying to say. Hopefully that was obvious. And then even smaller than the muscular arteries, we have these tiny arterioles. These are the smallest arteries that are going to lead directly into capillaries, capillary beds. We said capillaries are just a single layer of epithelial tissue, that simple squamous epithelial tissue. And if these are lining the blood vessel correctly, if there's not like a buildup of plaque or anything in the blood vessel, these things are very slippery and blood can move through them very easily. What else? There are a few different types of capillaries that we're going to see. The main type of capillary is highlighted down here at the bottom. This is called a continuous capillary. It's the same thing that we're looking at right here. These cells are all basically touching, but there's tiny little spaces in between those cells. We can't make it out on that slide, but this next one is kind of taking a transverse cut so that you can see the middle or the lumen of this capillary. You can see the single layer of cells that make up that capillary wall, and you can even see those little spaces that are going to allow things that are small enough just to squeeze through. That's what we refer to as diffusion, things that are just small enough kind of squeezing through. We'll talk about the forces that are going to allow things to either be pulled into the blood vessel or out of the blood vessel next time. Here we're just labeling types of capillaries. So here you can see the cell itself is intact. The only space is going to be in between the gaps between those cells. So continuous capillaries. I'll just jump to this one because that's where I was going next, and we'll jump back to the slides. Our continuous capillaries, you can see the only spaces for things to diffuse through are going to be those gaps in between the cells. But there are other capillaries in the body. The kidney, for example, when we get to the kidney, you can even see that. There's one of these models back here of, this is Bowman's capsule or the glomerular capsule. We'll see this when we get to the nephrons. And if you look at this capillary, it's the glomerulus inside this little Bowman's capsule. It's got tiny little pores on it. It looks like this. It's what we call a fenestrated capillary. Those pores are there on purpose, and they make it even easier for things to diffuse out of those blood vessels. It's like a sieve. And we'll see one of the functions of the kidney is to filter things out of the blood. Get rid of the waste, retain the things that we don't want to get rid of. So we see these leaky blood vessels in the kidneys. Okay, that's fine. If we look in the liver, there are capillaries that have even larger holes in their walls. These kind of look like Swiss cheese. They're called sinusoidal capillaries, and we'll see them when we draw liver capillaries. There's a model back here that shows liver tissue. So this is a model of some liver tissue. If we look inside liver tissue, it forms these little six-sided lobes that we're going to look at. And one of those structures is zoomed in where you can actually see the capillaries in between there. There are large holes in the walls of those capillaries. In the liver, that makes sense. One of the functions of the liver is to make plasma proteins, these big proteins that have to get into the bloodstream. And so these large holes allow those plasma proteins that are produced there in the liver a route into the bloodstream. Also, there's a lot of nutrients that is stored in the liver, so things have to be able to get out of those blood vessels and into those hepatocytes, the liver cells. So in some places, sinusoidal capillaries are normal, like in the liver. But just to kind of get on a side note, and to go back to this picture that I jumped over, here, if this link still works. Nope, let's try this other one. Oh God, I don't want to go down that rabbit hole. What you can see in this picture, it's a blood vessel in the eye. And you can see a single layer of red blood cells that have lined up just to squeeze through that narrow vessel. I had been saying the woes of being diabetic before because one of the main problems is that your red blood cells, they don't stay this disc shaped, and the thin little capillaries are just big enough to fit these red blood cells when they're in that flat disc shape. If they swell up like beach balls, then it's just going to shred those capillaries. It's going to tear holes in the walls of those capillaries. So what ended up looking like a fenestrated capillary ends up with much larger holes in it. It looks like a sinusoidal capillary. That's why people that are diabetic that have poked holes in their blood vessels, that kind of shredded their capillaries, especially in the kidneys, this is common. They end up having too much leaky fluid through those capillaries and have to go to the bathroom all the time because they're producing lots of urine. Okay, different types of capillaries. We've seen those. Let's see if I skipped over anything else. There were just a couple of pictures of capillaries, and we'll see as we get into capillary exchange in our next lecture. There are these tiny little sphincter muscles that lead into capillary beds. They can relax and allow blood to perfuse a particular bed of tissues into this capillary bed. So if you're in fight or flight, this is what would be going on in the walls of your heart or in your skeletal muscles. A lot of blood flow would fill those particular capillary beds, while at the same time, these little layers of smooth muscle would constrict and prevent blood flow from going to places like your digestive organs. And when we're in fight or flight or rest and digest, blood can't be everywhere. We have to kind of shunt it around to different capillary beds as it's needed. And here's just a close-up of that capillary exchange and kind of a preview of our lecture next time. These are just a bunch of continuous capillaries. I'll stay out of the way. But what you can see here, all of the blue arrows, is that me? Sorry about that. Turn that off. The blue arrows are just representing things being pushed out of the blood vessels. So just to kind of preview one of the things we will see, filtration, things leaking out of the blood vessels, is going to happen closer to the arterial end of a capillary bed. And then closer to the venule end over there, you can see the arrows are trying to indicate that excess fluid is being pulled into the veins. So this is like a vacuum pump over here. It's actually sucking in this extracellular fluid into the blood vessels. It's doing the opposite over here. We're going to see there's a pushing pressure that wins out closer to the arterial side, so fluid leaks out of these capillary beds. And then there's more of a pulling force that kind of wins out on the venule side, and you'll see more of this pulling of fluids into the blood vessels along that venule side. So we get into all of the details of capillary exchange. And I said before that not all of the fluid that leaks out of these capillaries is going to be picked up by the veins. So the excess fluid that doesn't get reclaimed by the veins would just float around in between these cells if it weren't for the lymphatic vessels. Those lymphatic vessels, their job is to collect that excess extracellular fluid that builds up, the stuff that leaks out of capillaries that is not picked up by the veins or the venule side. Okay, we've seen our capillaries. There's just some things about veins. We mentioned the smaller ones near the capillaries we're going to call venules. And then those venules are just going to lead into larger veins. Veins have a larger lumen, so we're going to see more blood in the veins than in the arteries. So about 65% of the volume of blood is going to be at any given time in the veins. And the largest of those veins, the superior and inferior vena cavas. Okay. So that's fine. There were a couple of things in the veins that were different from the arterial side, and these were all adaptations that the veins have. They're going to make it easier for blood to flow through them back towards the heart. There's no pump really on this side, so we're going to list a few things at least that the veins have that help blood keep moving through those vessels towards the heart and never back in the opposite direction. The first one we mentioned earlier, and those were the valves, those little flaps of the tunica interna that prevent backflow of blood. You see these, like I said, in places like the arms and the legs. If you have seen somebody with the varicose veins that you can see full of excess fluid, what happens in those cases is that these valves have become leaky, and you now have excess blood flow flowing backwards, and you can see that build up sometimes in the superficial veins. Another thing, in addition to those valves that kind of help the blood move in one direction or move through the veins, that is that these veins are typically going to be sandwiched in between skeletal muscles. And as your skeletal muscles contract, it's kind of like milking toothpaste through a tube. They kind of squeeze blood through those veins across the valves. So that helps push them from through one checkpoint to the next. It's called the muscular pump. It's another reason to go for a walk in the evenings. It's going to help circulate the blood. So we have a muscular pump. I don't know if it's mentioned here. I guess we just have it in this picture, but there is also what's called a respiratory pump. So as you take a deep breath like that, that expands the thoracic cavity, and that lowers the pressure in the thoracic cavity, and so blood will rush in to kind of fill that space. And so by taking in deep breaths, that's another thing that can help pull blood into that thoracic cavity. So we've got a muscle pump, we've got a respiratory pump, we've got valves in the veins, we've got the fact that those veins have a much larger lumen, so there's lower pressure there. So we've got a few things. This is kind of a super aerial view of different circulatory structures that we're going to look at, I guess is what I'm trying to say. We're going to see two circuits. We're going to see the pulmonary circuit, which is really just from the heart out to the lungs and back to the heart. That's the right ventricle, and one of the reasons the right ventricle was much thinner than the left ventricular wall. That left ventricle is going to have to pump blood all over the body. So above the heart and below the heart, even though that's not pictured exactly well there. But we'll see the systemic circuit is much longer, so that left side of the heart is much larger than the right side of the heart. We are about to see, I think in the next slide, some of the major blood vessels that we have on the lab list. And I was wrong. The next slides point out what I had mentioned earlier, at least in these bar graphs. I think this is helpful because it shows you in these larger arteries, the thickest layer is that tunica media, which we have that on the board, so we're fine. Even in these muscular arteries, you can see that tunica media, that layer of smooth muscle, is the thickest layer in these arteries. Even when we get down to the arterioles, there is still a significant layer of smooth muscle that's constantly determining by constriction or dilation whether blood flow is going to a particular capillary bed or not. Capillary beds, just a single layer of that tunica interna, very thin. This is where exchange is going to occur and our discussion of physiology next time. When we get to the other side of capillaries, into the veins, you can see right off the bat the thickest layer in the veins is always going to be the outermost layer, that tunica externa. I'll bring one of these things that we can list as well. There are a few of these vascular anastomoses that we will mention. This anastomosis is just a bunch of interconnected blood vessels that allow for alternate routes in case one particular capillary, the blood flow is prevented from going that direction. You have a lot of these around the knee, so if you kind of fold your knee up in one way, you wonder, is my foot going to get blood anymore? And there's a bunch of alternate ways for blood to flow around your knee down to the tibial artery and to the foot. So that's good, and that's one of the reasons your foot doesn't fall asleep as easily. And then the one that we are likely to have to label next time, because I like this model in lab, is the circle of Willis. It's another one of these anastomoses that we find inside the brain, and this allows for alternate routes for blood to flow to any particular lobe of the brain. So we will graph. I know this one is not a labeled picture, so we'll see a labeled picture of this circle of Willis, likely found on that model. I wonder if that's in the lab list. If not, we may have to label it. Now, I know this picture has got a lot going on. I won't read directly from this picture to you, but it's kind of a quick reference if you wanted to print this slide out. I'm sure the picture's in your book as well, of going through the lab list where you can look to try and find some of these arteries. What I am going to try and do in just a second is, it's just those two slides. I'll have those, maybe I can keep these up as reference when we go through the lab, and I'll erase this thing and put just a simple sketch of blood vessels as they leave the heart. The one thing that I want to draw, I guess, about these major arteries and major veins is how they branch differently, how the arteries and veins branch differently right around the base of the heart. You can't see it very well from this picture, but we're talking about the base of the heart. Remember, the heart's like an upside down pyramid, so the apex is down at the bottom, right on top of the diaphragm, and all the attachment points where all the blood vessels enter and exit are up there at the top. So that's our base of the heart. I kind of want to draw a couple of those. And then we're going to get into a heart dissection after a short little break. Is everybody good with that so far? Do you need that? We're going to find these blood vessels in lab, and I try and point to them, but it's even sometimes hard to see on the models. So I'll start this way first. I haven't drawn this in a while, but I'll try and fit it over here. I'll start with the heart, and I'm just going to put a dotted line right here and let that represent the diaphragm. And I guess we can start with arteries. So the thing that you typically see on that heart model is this aorta that comes out. It does its aortic arch, and then it drops down below the diaphragm into this abdominal area. When we get down here, we're going to see it fork into these common iliac arteries and eventually down into the leg. We'll see internal and external iliacs. It's really this external iliac that extends down into the leg. So maybe I'll put a couple of dotted lines down here that represent the groin area and where we go down into the leg. I'm not going to be able to draw too much of the appendages on this, but I just wanted to draw really how the blood vessels branch around the heart. This aortic arch, to be clear, and I don't want to crowd this drawing up too much, the first part here is what we're going to call the ascending aorta, where blood is going up. You've got the arch itself, and then the rest of this is going to be the descending aorta. And as it descends, while it remains in this thoracic aorta, it can sometimes be referred to as thoracic aorta above the diaphragm, and then abdominal aorta below the diaphragm. But you have on your list these branches that come off of the aorta. We have to know the three branches. One is, the middle one is going to go right up into the neck. The far one here on the left is going to all by itself go out into the left arm. And then the first one that we see, I'm going to try and draw that a little bit thicker than the other ones. This is what we call the brachiocephalic trunk, and I'll try and label this in a second. This brachiocephalic trunk is going to branch into what we're going to call a common carotid and a right subclavian. So let me attempt to label these things. We obviously have our aorta right there. The first branch off of that aorta, this thing is our brachiocephalic trunk. And that brachiocephalic trunk is going to branch into this first thing that we're going to call the right subclavian. So right subclavian artery. Over here, kind of all by itself, is the left subclavian artery. I might be writing kind of small here, but hopefully you can come closer maybe and take a picture if you need to do that. The middle two that we have yet to label, this one that is coming off all by itself, branching off of the aortic arch in the middle, is what we're going to call the left common carotid. There's a lot of places where arteries and veins are going to get called the same thing, like common iliac artery. We're going to see a common iliac vein. Up here in the neck, the aorta, the arteries, I should say, are going to be called carotid. So there is a left common carotid, and this other branch that branched off of the brachiocephalic trunk on the right side is going to be the right common carotid artery. And it's true, if we could zoom into this, we would see that if we follow those common carotid arteries up far enough, they're going to branch into an internal carotid and then the external carotid. Internal carotid and external carotid. We'll see these internal carotid arteries when we draw that circle of Willis. 80% of the blood that flows to the brain flows through those internal carotid arteries. Anyway, I'm running too far off the board where I can label that any further. But I wanted to compare that to how the veins branch around the heart. So maybe I can squeeze another heart in here, and I'll put a dotted line that once again just represents the diaphragm. I'll try and get my body out of the way, and with a blue color, let's label some of these veins. So from below the heart, we said the blood's going to come in through this inferior vena cava. So blood is going back towards the heart through this inferior vena cava. Blood's also going to be coming towards the heart by way of our superior vena cava. And if we were to go down here, this inferior vena cava, like I said, we would see a similar split like we see with the arteries, where there is a common iliac vein, an internal and external iliac vein, and then the external iliac veins go down into the leg. We'll call them the femoral veins. So a lot of time there are similarities, but above the heart with the arteries and veins, there is a little bit of a difference. So I'm going to get those arrows out of the way just to avoid a little confusion. And right here above the heart, this superior vena cava is actually going to have kind of a symmetrical fork there. And I'm going to draw that superior vena cava just a little bit smaller so that I have room to label this. So that little bit there is our superior vena cava. And above that superior vena cava, we can see branches in either direction. Both of those branches are going to have a symmetrical fork that goes out like this. And the ones that are making their way up into the neck, we're going to call these the common jugular veins. We've got our superior vena cava that goes up. The superior vena cava is going to lead, I barely left myself enough room there. Maybe I can give myself a little bit more of a labeling space. That vessel right here is on the left side. This is going to be our left brachiocephalic vein. So in arteries, just to make sure there's a difference there, there is a brachiocephalic trunk in the arteries, and that brachiocephalic trunk branches into the right subclavian and right common carotid artery. But in the vein world over here, there is its own right brachiocephalic vein. I barely left myself enough room there. Let me move superior vena cava down some. So we have a right and a left brachiocephalic vein. And if we follow either one of those brachiocephalic veins out, let's just follow the one on the left side, for example, it's going to branch into this left subclavian vein and left common jugular vein. This one would be our left subclavian. And obviously over here, this would be our right jugular, right common jugular. And if we followed these jugulars up, we would see them split as well. Let me label this right subclavian vein. And I'm going to make one correction to that, actually. I'm going to come in here and instead of calling this common jugular, it's another one of the differences between arteries and veins. And this is where I'm going to get the light color blue. We'll do this when we get into veins. Veins can get different names depending on whether they are deep or whether they are more superficial. So just after this first branch, this is, instead of calling it a common jugular, we're going to call it internal jugular. So right and left internal jugular. Just to the lateral sides of those, I'm going to put it here in a little blue squiggly line. Those are going to be what we call external jugular veins. I'm not going to label them here. It'll make it too congested of a labeling. But there is a model, one of those, maybe it's one of those torso models back there. We'll hunt this down in lab. We'll find an internal and external jugular vein. External jugular veins are closer. They lead into the subclavian veins, and then into the brachiocephalic veins, into the superior vena cava, and eventually into that right atrium. So one brachiocephalic trunk in the arteries, two brachiocephalic veins in the veins. And for the rest of the stuff down into the legs, we'll see some of that on the models. So with my one correction, that's at least the distinction between the branching pattern of those major blood vessels above the heart.